We're here in St Gallen at the Stiftsbibliothek, and we're here with the Inks and Skins team, and they're going to use some exciting scientific techniques to try and unlock some of the secrets that are hidden in the manuscripts.
This is probably one of the most exciting things I've ever done. To have these books together in one library is a remarkable thing. Unique artifacts. Every time we turn a new leaf, we're doing something that no one has ever done before for Irish manuscripts.
There's a question often asked by the great scholars about whether books were produced here by Irish monks or Irish people, or whether they were brought here by monks travelling over. Now we're engaging with science, which gives us a much more definitive proof of where something might be made.
So this is our XRF machine. It stands for X-ray fluorescent spectrometer. So it's using the technique of spectrometry to analyze what elements are present in the manuscript, both in the inks, the pigments, and the parchment itself. There's an X-ray laser, essentially. It doesn't harm the book at all. This is a non-invasive technique. So then our machine, we're able to see in real time, as we're scanning, which elements are present in that specific spot. We're able to see if I get a spectra that has a very high count of mercury, we know that the chemical composition of vermilion is mercury-based. So that allows us to identify which pigment they were using for the red.
What really interests me is the things that we don't know anything about, namely the recipes for the ink, recipes for the pigment, the way the books were prepared prior to the application of ink or pigment to them.
So one of the techniques we've been using, and this is a version of it, is multispectral imaging. That means that we can capture information not just in the visible, what we see with our naked eye, but outside in the ultraviolet and the infrared. Sometimes particularly the organic pigments, things that are made from plants, can be indigenous, so they can come from that local region. And they're a lot harder to identify, so that the little spectrophotometer I've been using, that helps us understand potentially what that plant-based pigment might be. Are they from the time period we expect? Are they from the place we're in? Or have they travelled from other countries?
So one of the unique aspects of this visit in preparation for the exhibition is the collecting of DNA sampling. So this is genetic fingerprint on the material itself. What that allows us to do is identify the animal that these manuscripts have made of. We've got to remember that these early medieval manuscripts are all produced on skins, and they're the skins of an animal. We can gather samples from the surface by rubbing. We collect that in a vial, and then the genetic code can be extracted from this, and it will give us information such as the type of animal. And as we build up this database, we may even start to be able to include things like the geographic location of where the animal was.
Here in the genetics department in Trinity College, for quite a few years now, we've been working on the ancient DNA of cattle. And why do you study cattle? Well, because cattle are important, but also they tell you something about the human past.
So here I'm going to extract DNA from the St Gallen manuscripts. And this is an example of the rubbings you get from the manuscript. So I'm just adding the extraction buffer to the rubbings.
It's amazing, it's almost homeopathic, but you get enough DNA from that to be able to reconstruct the genome of an animal very, very often.
So this liquid is now ready for us to process and send it for sequencing.
We successfully sequenced a good amount of DNA from 3 manuscripts. And when we did that, I compared it to a database of ancient genomes of cattle from Britain, Ireland, and the continent. We were able to see that they were much more similar to those from Ireland than they were from the other 2 places. These manuscripts were Irish in origin. The animals that they're written on were Irish cattle.
So we're really grateful to our lenders in the Abbey of St Gall for the opportunity to get access to their manuscripts during the course of preparation. It's been a really unique opportunity to use scientific techniques during this exhibition because they've been able to uncover actually new findings about the manuscripts.
We felt we were with something very familiar, having worked on similar manuscripts in Ireland. And in fact, the research results confirmed that. It's really wonderful to think that over 1,000 years after they made their journey eastwards, they've made a similar journey, a return journey back to Ireland for this wonderful exhibition. And for us to be part of that and to be part of the scientific backup for that is just unspeakably an honour.